So looking at figure two, we see that they're looking at two different examples of an actually edited genome in in the human pluripotent stem cells. So they look at two examples of polymorphism and a point mutation that they induce. In the first case here, it's a C to T mutant in exon 6, and then in this case on the right, it's an A to C mutation on exon 8 of the same gene, the CHR11 gene. So anyway, in terms of the selection effects in both cases, they observed that with increased pyromycin, the number of colonies decreased, which is expected because not all cells are going to be resistant or take up the the plasmid. So with that in mind, the cells that did take up the plasmid and were resistant to pyromycin seem to have a high gene editing outcome, and more importantly, a high ratio of HDR to indel, than which is what the whole objective of the study, one of the main objectives of the study was looking for, basically was to find a method to increase the preference for HDR, which is the correct form of gene editing in most cases. And in the right, you can see the same trend: pyromycin increase with a decrease in colonies, and also an increase in an overall increase in gene editing, as well as a significant ratio of HDR to insertion deletion. This component here, D and F, compares this process of stringent selection of pyromycin to another option that other studies have used, which is GFP, or fluorescent selection, using FACS, or fluorescent cell sorting, which we have discussed in the lecture before, where they basically sell an individual line of cells, and then cell by cell, based on the fluorescence, they're sorted. So in this case, you see that cells that express GFP are much less significant as when GFP is highest. So the number of colonies in the well are the highest when GFP is lowest. So in that case, you can also see from these data that there's a lot less overall colonies than in this case, which suggests that FACS is detrimental to cells or damaging to cells, because there's a lot less number of colonies in each well that are measured with this technique. And that goes the same with overall percents of gene editing. It's also decreased, which is another key finding, is that pyromycin seems to be preferred in general compared to standard fluorescence techniques. So figure 3 is just another way of observing the data that we saw in figure 2, with overall editing increasing with pyromycin selection. So higher pyromycin resulted in overall increase in percentage of cells with that have been edited either by insertion deletion or HDR. And this is not to say that if you increase pyromycin, then gene editing will increase in general or be more efficient. No, it's basically saying that because pyromycin is increased, there are more cells that there are less cells that can survive if they don't have the pyromycin resistance gene expressed. So that just means that this just means that with higher pyromycin, the total percentage of cells that are in final culture have increased, or higher percentage of cells of those final cells have been successfully edited with either insertion deletion or homology directed repair. So that was the kind of the first half of the paper that I discussed. And before we go to the second half, and especially the second half of the figures that Brycelyn is going to discuss mainly, I wanted to introduce a couple more, review a couple more topics that are important to understand the figures. The first being Sanger sequencing. So Sanger sequencing is used to sequence these polymorphisms or point mutations at the end and kind of compare results. So how does Sanger sequencing work? As we discussed in literature, the key components are primer, template, the DNTPs, and deoxy DNTPs. So deoxy DNTPs are going to have a fluorescent tag associated with them. There's going to be 4 different fluorescent tags, and deoxy DNTPs terminate the sequence because there is no oxygen, there is no hydroxyl group necessary for continued polymerization of the DNA onto the template from the primer. So with that being the case, these are going to be separated or based off of their size through. In the old days, it was electron spectroscopy, but now it's more high HPLC, high precision chromatography. So this is what it looks like here on the right, the different peaks and indicating in succession the sequence of the peptide, or the DNA sequence, not peptide, sorry. And then the next thing that's discussed in the following figures is next generation sequencing, which, as we know from lecture, basically in summary uses techniques for sequencing different small fragments of DNA of interest that has been cut up, and then attached to a flow cell where these are successively conjugated and cleaved in different lengths, and so they can be observed by fluorescence spectroscopy in this kind of format, where each of those fragments can be sequenced, and in bioinformatics, gene sequencing or sequencing data is mapped to those sequences to predict the sequence of interest, whether it be RNA, DNA, or whatever it may be. But basically, an important thing to understand from gene sequencing is that it uses the fact that we know the genomes of humans as well as other organisms to kind of predict a quick fashion small cut up sequences from a sequence of interest to predict the locations of different genes and such. It's a very high throughput method. It's a lot faster and high throughput than Sanger sequencing. And with that, Brycelyn would carry on for the remainder of the figures as well as our proposed experiment. So please stay tuned for that. Thank you very much.